Hello, everyone. Greeting to all and welcome to today's webinar for the GSK Biomanufacturing Omics Data Challenge brought to you by 9 Sigma. My name is Serena Bess from 9 Sigma, and I will be your host and moderator today. Please note that this webinar is being recorded, and the recording and transcription will be made available on the GSK Biomanufacturing Omics Data Challenge page on 9 sites. For the agenda today, so here what we can review after I introduce our speakers. They will discuss the project in greater depth, including JSK's initiatives and the topic for the challenge. Then we'll take some time addressing to your questions during a live Q&A session, followed by a brief summary of project information, including how to request additional information or assistance. This is a reminder to all participants are in listen mode only. As we proceed through today's presentation, please feel free to ask questions at any time using the question box through your GoToWebinar control panel located near the top right side of your screen. We'll keep track of your questions and respond to them during the Q&A portion of this webinar. Should you need any information or assistance outside of today's webinar, please contact 9 Sigma provider help desk at phd@9sigma.com. Yeah. And at this time, I would like to introduce our panelists for today's webinar. We'd like to welcome Julia and Rui from JSK, as well as Stephen from 9 Sigma. And thank you for everyone that has joined us today for this webinar. And at this time, Julia will tell us more about JSK and introduce their innovation challenge. Julia?
Thank you, Serene. Hello, everybody. Hopefully you maybe have heard of GSK before. We are a global healthcare company with a purpose to help people do more, feel better, and live longer, with a very strong focus on science and innovation for our medicines, our vaccines, and our consumer health products. Next slide. We operate globally everywhere around the world. You can see a few figures here just showing the scale of the company, and also you can see as well we have a very strong focus on innovation, and so we have we look for a fraction of our sales, a large fraction, more than half now, come from our new innovative products. And we also have very strong focus on global health. We have, you can see there, we contribute globally to a lot of community initiatives around global health, and we even have a division of the company focused on global health challenges. Next slide. Rui and I come specifically from GSK Vaccines. Developing in GSK Vaccines, we develop vaccines for all stages of life. You'll see here a picture of the herpes zoster virus that causes shingles, which we attack with our famous Shingrix vaccine. And just a couple of brief figures there. Vaccines have been historically the second most impactful intervention for human health in history. The only thing that has saved more lives than vaccines is clean water. And also GSK estimates that around 40% of the children in the world get at least one vaccine from GSK. So we are very proud of our global reach of our vaccines and the positive impact we make on health with that. Next slide. So this challenge specifically, GSK, we produce protein-based vaccines, among other kinds, through a cell-based bioprocess, very similar to any other process used commercially to make protein-based vaccines and therapeutics. That process is very expensive, it's complex, it's time-consuming to carry out, and at a commercial scale, small improvements in the efficiency of production there can have a significant positive impact on the process, on the yield, and so we're quite interested in how we can always make our processes better. And we have lots of quality issues show up things like truncation, frame shift, mutations in the protein aggregation, all of these things commonly can occur in fermentation processes. So we at GSK conducted a set of experiments on fusion proteins that we were producing that were under investigation at the time for the treatment of cancer and HIV. And we were trying to better understand the causes of these common issues in production and how we could prevent them from occurring by changing the conditions of the fermentation, or if there was something else we could change to prevent that from happening. Next slide. So why are we doing this challenge? We, of course, have analyzed our data set ourselves, but we are quite interested, very interested in what other researchers might be able to do with this, what other insights could be found in this data through some sort of analysis that we haven't thought about before. We're very interested in whether there are new data analysis methods that, again, that we haven't maybe used before, we haven't thought of using on this kind of data. And we're very interested in having a collaborative exchange with the winners. So we will ask that the winners, after they've had a chance to look at the data, do their analysis on it, we'll have a presentation with representatives of the Vaccines Technical Research and Development Department, where we can discuss the results they had, their applications. We're very interested as well in potentially extending that collaboration further if there are promising applications for GSK. And we're also interested in this in the spirit of open science. We know that access to data sets, especially industrially relevant data sets like this, can often be a bottleneck in bioinformatics research. So we would like to make this data set available to almost all of the submitters. So if there are reasonable proposals from reputable researchers, the ones that are not selected as the winners of the challenge will still be offered the chance to access this data set and analyze it and use it as they wish. So we're hoping that this data set itself, we think can be a significant contribution to the research community. And we're also interested in demonstrating to our internal GSK management that there is interest in the scientific community in access to this kind of data, so that in the future, we maybe can convince them that there are other data sets like this from GSK that we don't need to protect anymore, that we could similarly make available. So if we have a strong response here, that will hopefully help us make that case in the future. And now I'm going to pass this over to Rui, who is going to tell you about the technical details of the data set.
Thank you very much, Julia, for a very clear description of this challenge background. And I hope that you can hear me fine. I'll now shift towards a more detailed view of the proposed challenge, a scientific problem, and also available data set. The problem that we present here is very closely related with the GSK core business, the production of proteins that will function as antigens in our vaccines. So in this particular case, as Julia just mentioned, we focus on two proteins, so WT1 and F4CO, that were designed for the treatment of cancer and HIV respectively. These two proteins were produced in an E. coli strain that contains few genetic modifications compared to other strains classically used in academia. So these mutations don't affect the central carbon metabolism for those of you interested in this type of analysis. These two proteins that were used as case studies here were selected given that the range of quality issues, titers, molecular weight, and complexity that they present. It is also important to refer that this type of production related quality issues are very relevant for us, as they contributed to the stop of several projects. So it's very important for us to understand the mechanisms underlying them and to be able to solve them for any other future antigens that will present similar issues. In addition, these two strains used for the production of these proteins are genetically equivalent, with the exception of the plasmid that contains the gene of interest. Next slide, please. So to be able to compare and generalize the learnings from this exercise, we ran a series of equivalent production processes from these strains. These were performed at 20-liter scale, which is the production scale of some of our vaccine that focus on a less prevalent condition. This is, of course, excluding some of GSK blockbusters that are produced at larger scale. As such, this data set might be very interesting as a way to connect academia and industry, as Juliet previously mentioned. The process itself can be described as a classical fed batch process following the usual inoculum preparation in a pre-culture flask from a frozen seed. This is then transferred to the referred 20-liter bioreactor, where a batch phase precedes fed batch that is started when glucose is depleted. Further, when an optical density of 80 is achieved, the induction starts, and this is achieved by the addition of IPTG. So as mentioned, this is a plasmid-based expression system based on the LAC operon, so quite comparable to other applications in academia. In addition, we should mention that a chemical defined medium is used across this production process with antibiotic as a resistant marker. During the whole process, the usual parameters are also measured and controlled, namely temperature, agitation, pH, and optical density. With the high cell density in this E. coli production process being one of the key differences when compared to other non-industrial strains and bioreactor conditions. Next slide, please. Overall, 14 processes were run and characterized in detail, 6 of which for F4CO and 8 for WT1. In these 14 processes, we followed different sampling strategies depending on the scientific questions we tried to answer with the particular set of conditions. So this, to the best of my knowledge, is a data set that represents the most complete characterization of any industrial upstream production process, covering virtually all possible omic layers. And even at GSK, this required a collaboration between several groups, 5, if I'm not mistaken, and other external collaborators. Given that simultaneously we needed a broad and specialized set of expertises that were required, of course, to produce, to treat, to analyze, and to merge these different data sets. So if we go more in detail, the data set is composed by the usual readouts from a bioreactor, as I mentioned. These can be achieved on and at line. These include, of course, pH, oxygen, temperature, agitation, biomass in the form of optical density, and also dry and wet cell weight and CFUs. Such biomass was also characterized in terms of amino acid, lipids, and genetic material. So regarding the omic data sets, at the metabolic level, both internal and external metabolites were quantified, and the most common ones were labeled. Normally, the full data set will, with unidentified metabolites, will also be shared with the selected participants. Regarding the transcriptomic dataset, this was produced with the Illumina sequencer, targeting the gene expression fold changes after induction. So this was used as the normalization sample. The quantification of tRNA was based on microarray technology. So the experiment design included a complex combination of fluorescent dyes for each of the samples and time points. And by this, I mean that each sample was labeled with at least one dye at once. And we also combined the antigens that resulted from different tRNA levels so that we could more directly compare the results that were measured with these microarrays. In addition, we have also determined both total and charged tRNA levels so that we can get a more complete picture of what is happening in the cell regarding the tRNA, the translation process. For the proteomic dataset, it was run after a mild protein purification and targeted the characterization of the protein of interest and respective truncation variants, given that this is the main objective of this project. Lastly, we will also be sharing with a very unusual data set, especially for industrial processes, that is the ribosome profiling. This was only run as a proof of concept. So we have 2 samples for 1 fermentation that were running in triplicates, as this experiment for bacterial samples presents some additional technical challenge when compared to eukaryotic systems. Well, lastly, to stress again, to the best of my knowledge, this is the most detailed characterization of an industrial production process and could be fundamental to facilitate the application of academic discoveries in terms of methodology and models to industrial settings. Next slide, please. So as such, we are very excited about this challenge, and we are definitely looking forward receiving your proposal and ultimately collaborating with you to further understand the biological mechanisms of E. coli, especially those happening in our production processes. In particularly, we are very interested in innovative modeling approaches that integrate data across different layers to generate new insights, as this represents one of the active research fields, and we have reasons to believe that this data set can be fundamental to drive the development and the standardization of this area, especially again in terms of transition from academia to industry. The submitted proposals will be evaluated by a panel of GSK subject matter experts based on the uniqueness and innovation of the approach and how it combines the different data sets, as well as how relevant the potential conclusions can be to GSK. One note that should be stressed is that we are not targeting the combination of all the data sets. Is also completely fine to combine only 2 or 3 of the data sets that are of interest for a particular research group. Additionally, we also expect the team to be knowledgeable in the area of the submitted proposal, hence being able to execute it. Lastly, it should be noted that the proposals that are not selected as winners will be evaluated for data access with special emphasis on educational uses of this data set. So thank you very much, and back to you, Stephen.
Thanks very much for that, Rui. I'd like to tell everyone about how to submit a proposal to the challenge. First of all, please visit the challenge website, and the link is 9sigma.com/s/GSK Biomanufacturing Challenge. And there you'll find a lot of information about the challenge, about the data set, the challenge timelines, and the types of proposals that GSK are interested in, the types of proposals that are going to excite GSK. All of the key information is shown on the challenge page. Plus, at the bottom of the page, there are 2 attachments that give you even more information. When you're ready to start writing your proposal, please click the big orange respond button at the top of the page. If you've got a 9sites account already, you'll be asked to log in. If you don't have a 9sites account, you'll be asked to create one. And that's really easy to do. You just enter your email and create a password. And that just means we can contact you again about your proposal. And then you'll see the response form. And this on the right-hand side here shows the response form. It's really just a series of boxes where you type in your response, or you can copy and paste it from another document. There's space for you to upload any attachments, and that could be additional diagrams or images, publications, your CVs. Some people actually write their whole response in a Word document or PDF with all their answers to all the sections, and upload that as an attachment. And you're welcome to do that if that's easier. You can save the response at any time and come back to it later. And when you're ready, you can click the Submit button. So what should you include? Well, the first thing to say is we don't want you to include any confidential information. There's no need to disclose your special know-how or your proprietary knowledge. You can tell us what you can achieve, what types of outcomes you can deliver, but there's no need to tell us exactly how you do it if you don't want to. The second important thing to include is that GSK are really keen to understand what approach you will take. What's unique or different about your approach? And how could that lead to the creation of new, interesting models that integrate the data in new ways to allow you to get new insights from the data? And you'll see in the response form, there are sections where you're specifically asked to describe this. So please include this information. Finally, we'd like to know about you, your experience in data analysis, details about your team, your capabilities. And when you've completed your response form, you just need to check a checkbox showing you that you agree to the terms and conditions, and then click on the Submit button. And the response will be automatically saved in our database. And just one final reminder, the deadline for responses is the 14th of October at 5 p.m. Eastern US time. That's everything I wanted to say about submitting a challenge entry, and I'll pass back to Julia to talk about the awards.
Thank you, Stephen. So the award, there will be 3 winners selected, each for an award of 7,000 euros. And additionally, we would ask the winners to present their analysis to the GSK Vaccines Technical R&D Department so that we can then discuss your analysis, its application. Hopefully also we could discuss further collaboration and how GSK may be able to fund additional analysis and additional work with you if the initial analysis has good applicability to our problems at GSK. And again, we want to stress as well that proposals from reputable submitters who are not selected as winners will still be able to access the data set if they would like. And we especially encourage proposals for purposes like teaching. And if I'd also like to say if you are only interested in the data access and not really concerned about trying to compete for the award, you don't have to put a whole lot of detail into that proposal. You can just give us a very brief explanation of, how you would like to use this. What kind of students are you thinking of using this with? Who are you, so we can make sure that that you are not somebody that presents a conflict of interest or would otherwise be problematic for GSK. But in general, really any researchers who would like to have access to this data set, we are very eager to encourage that. And now I believe I will hand it to Cyrene for the FAQs.
Yeah, thank you, Julia, and thank you, Stephen and Ryozel for the information. So now we have like a couple of FAQs here. These questions kind of will be answered by our panelists. So the first one, will GSK claim IP on the analysis, Julia?
Yes, so first I should say I am not an expert in IP in any way. If you do have further questions on this, I'm happy to connect you. We can connect with someone who is, so just use the question email at 9 Sigma, but the general answer for this is GSK is not seeking ownership or exclusivity of any intellectual property generated through this challenge. So submitters are free to seek your own IP protection from your solution, but we will ask for knowledge sharing from the winners. So an explanation of your methodologies, copies of your custom software if appropriate, and a corresponding non-exclusive license to that IP so that GSK could continue to use it ourselves. And if you'd like to see more on this, all of the relevant terms are in the challenge terms and conditions. So that include the challenge terms and conditions include all of the terms and conditions for data access. There's not going to be anything additional to that we ask you to agree to when you access the data.
Great. Thank you, Julia. Now someone asks, where is the description of the data set, Stephen?
Yeah, so if you go to the challenge website, at the bottom of the page, there are two attachments. The first one is the terms and conditions that Julia just mentioned, and the second one is a description of the data set, and that includes all of the information that Rui talked about earlier and more information. And you can access it just by using that link, 9sigma.com/s/1a, or by going to the challenge site and opening the attachment at the bottom of the page.
Yeah, just to be clear, as it says here, all the information that is submitted should be non-confidential.
Yes, absolutely. We're not interested in getting confidential information from you. That's really so that if you do want to patent or protect your inventions, you can still do that. So please don't submit non-confidential information. We'd love an explanation of what you can do, what you believe you can achieve using your approach, but there's no need to tell us exactly how you do it if you don't want to.
Great, thank you, Stephen. Also, another frequently asked question is, can I access, the data without submitting a proposal? Julia?
No, unfortunately, you cannot access the data. But if all you are interested in is data access and you're not trying to compete for the award, as I said, you don't need a very detailed proposal for that, just enough for us to be confident that you're a reputable researcher, basically that it wouldn't somehow embarrass GSK if it became known that we had given you this data set. So if you're working in research and you have some idea of what you'd like to do with this, but you don't care about winning the prize, you can just give us a very brief description of that.
Okay. Also, can I publish my analysis, Julia? So someone is asking if they can publish the analysis.
Yes, you absolutely can publish your analysis. We do. Yes, sorry, bit of an audio issue there. Yes, you can publish the analysis. This is in the terms and conditions as well. We just ask that you acknowledge that you got your data from GSK, and we have a specific code we'd like you to include, just so that we can, keep a search alert so we know when people are publishing about this. Again, largely just so we can sort of demonstrate back to our management that this is contributing positively to the scientific community, we hope. But you absolutely can publish.
Okay, thank you. Thanks, Julia. Thanks, Stephen. Next slide, please. So now we'll open up the session to questions from the audience. Please continue to submit your questions through your Q&A chat box. This is an excellent opportunity for you to pose your questions directly to our speakers. Again, to open the question box, you first have to open your GoToWebinar control panel if you haven't already. So yeah, next slide, Stephen. Yeah, and here we go. So someone is asking here, how long should my proposal be, Stephen?
Yes, of course. So there's no limit to how long the proposal should be. You can make the proposal as long as you like, but generally we find that short and interesting proposals are often more successful than very, very long ones. So typically, if we look across all of the projects that we run, successful proposals normally contain about 1,500 to 1,800 words. Most of that is often in the description of the proposed technical approach section. We're also asking what's unique about your approach. So here we're suggesting perhaps about 200 to 250 words. And again, we're asking for a description of your team, and typically this is perhaps 150 words long. But don't forget, if you already have a great description of your team or the CVs of your principal investigators, you can add those as attachments to the proposal, and you don't need to write anything new.
Yeah, thank you. The next question, someone asks, can we submit as a team of people? Is there a limit on the number of people on the team? Julia?
Yes, absolutely. You can be a team. You can be a team of as many people as you would like to be. We do need there to be one lead person who will be, if selected from the award, we will be sending the award to one person, and then how you would like to distribute it amongst yourselves is totally up to you. But absolutely, you can be a team. And if it is a team, again, you don't necessarily have to give us a CV of everybody. If you'd like to give us, the CV of the lead researcher or the lead couple of people, and then, include that there is another team working on this as well, that's fine. We don't need, full information about absolutely everybody if you're a big team.
Yeah, thanks, Julia. Also, there is another question. Were there any off-gas monitoring like O2 and CO2 in exhausted gases measured? Then did you measure the inlet gas flow rates, airflow and oxygen flow in SL, PM, or mass gram per minute? Rui?
Yes, thank you very much for this question. So in terms of gas monitoring, this was indeed measured, except for one or two fermentations, if I'm not mistaken, that had some problems with the measurement. But the remaining 12, this data is of good quality and available. In terms of inlet gas, this is not controlled. So this is only controlled in terms of dissolved oxygen.
Okay, another question, Rui. Someone is asking, how about pressure measurement inside the bioreactor? Would it also be available in dataset?
Yes, so indeed the pressure was also measured and controlled to some extent in the bioreactor. And normally these data will also be shared with the participants, at least the selected ones, of course.
Yeah. Someone else is asking, is there a page limit on the length of the proposal? But I think we've answered that, Stephen, right?
No, there's no page limit.
Okay.
You can submit as much as you like. But again, we do find that proposals that are very clear and concise are often more successful than proposals that are very, very long.
Okay. Also, someone else is asking, are we allowed to use a non-GSK data set in tandem with the GSK data set for the models? Julia?
Yeah, if you have a non-GSK data set that you think you could combine with this one, that would be very interesting. If it's proprietary to somebody else, especially if it's proprietary to one of our competitors, that might be a problem. But if it's something publicly available, that would certainly be extremely interesting.
Cool. Yeah. Maybe also, Julia, someone also is asking, is there any specific question you are looking to find the answers for?
The overarching question is, are there things we could do in our fermentation process that would let us avoid some of these production problems, or could we somehow predict that they're going to start happening, for example? But anything else that gives us more information about our process would be very interesting. Our goal as the technical R&D department is to develop a robust and scalable and repeatable process that gets handed off to manufacturing when a vaccine becomes commercial. So anything that would help us help manufacturing produce the proteins in a repeatable way would be quite interesting to us.
Thank you. Thank you, Julia, for clarifying that. There is also someone else asking, is the data in its original instrument format, or is it transformed to facilitate access? Rui?
Yep, that's indeed a very good question, because we are talking about a wide range of instruments and data formats that were generated in this project. So normally the data that will be shared with the participants is already pre-treated. So for instance, in the transcriptomic data set, we are talking about sharing with the selected participants the fold change of each of the genes. Likewise, for the metabolome data set, we are thinking of sharing already the quantified well, targeted and untargeted experiments, not necessarily the peaks or the FASTQ files, also because this involves an additional problem in terms of sharing the data set and which system we would be using. However, if there's a rationale behind a particular use of the data set, we are definitely willing to evaluate if it's okay and feasible and interesting for GSK to share a particular specific data format.
Yeah, thanks, Rui. Also, a lot of questions are coming in. So another question: are the products secreted out? Rui?
So indeed, this is a possibility, and it happens in some of the proteins. This is not the case for the WT1 and F4Co. There's a minor presence of these proteins on the periplasm, but again, this is minor. It's not really significant.
Okay. Also, another question: what's the ultimate aim of the experiment? Someone is asking. Julia, maybe?
Yeah, I mean, I sort of answered similarly before. The ultimate aim of this study was to help us figure out how to better make our proteins. How could we facilitate through the control of conditions in the bioreactor, can we influence these production quality issues? So that was the initial aim. If somebody thinks they can also get some other insights that would be relevant to GSK, we would be extremely interested in hearing that. Rui, do you have anything to add? I think you were in the team that initially got this approved when we started collecting all this data.
Indeed, I make your words mine. So the overall objective of this particular project is indeed that, so improve the quality of the protein produced, facilitating the downstream processes that we also develop at GSK. I would perhaps extend, if there is some participants willing to target other objectives. We are certainly interested in reducing the fermentation timelines, improving the titers of the fermentations. But again, this is not strictly speaking the objective of the of this particular project.
Okay. Also, another question, someone asks, if based on your experience, are there multiple mutated proteins in the same batch? If so, how many batches are profiled by MS species? Rui?
So regarding the mass spec dataset, this dataset was targeted for the protein of interest, so F4Co and WT1. In this case, we see some, well, to some degree, some amino acid misincorporation, but in terms of mutations, so the genetic background of the E. coli strain that we are using is quite stable. However, at the transcriptomic level, we see some minor minor mutation, minor errors during transcription as expected. But these, as we have found, they don't explain the overall quantity of misincorporations and not the nature of it also.
Yeah, thank you, Rui. Also, there is another question asking about the timeline. So is there a timeline to report the results of the analysis? Julia, maybe, or Rui?
Yeah. So we don't have a specific timeline. We would expect this presentation to the GSK Technical R&D Department. We will, mutually agree on a time that works for all of us. We would probably hope to hear at least something in, within a year. Of course, you can keep access to the data set and keep using it and analyzing it, as long as you want. But we would like to get an update at least within a year.
Okay, great. Also, someone else is asking, are there any data for the product quality of interest? Rui?
So the product quality was assessed internally, been using some of the common analytical methods, but I would say that the main method used to assess this quality was the mass spec data. I think this is the most the highest definition that we can get from this type of samples, and that we will be sharing with the participants.
Okay, thank you, Rui. Also, another question regarding the challenge. When are the winners going to be announced? Stephen?
Sure. So we plan to announce the winners and notify everybody about data access in December of this year. We haven't fixed a date for that at the moment, but everybody will be informed in December.
Yeah. Also, regarding the data, product quality, another question. Are there any data for the product quality of interest? I think we've answered this, but I'm not sure. Rui?
I think we have answered. So
yes.
Again, the product quality was measured by mass spec, so comparing the amino acid sequence to the expected one. And additionally to this, we also have some SDS page data that we might be able to share that show some an unexpected pattern in terms of protein weight.
Yeah, clear. Another question, looking at the table of conditions, what was the reference for the fermentation experiments? Rui?
Yeah, thank you for that question. So the reference, it actually depends on what are you focusing on. So for instance, for the transcriptomic and ribosome profiling experiments, the reference was a sample just before induction. And in terms of overall process, the reference fermentation was a process that was developed or that was established as being more similar to other processes already being used in other projects at GSK. So that was our benchmark.
Yeah, thank you, Rui. Someone else asked, beside endpoint product quality assessment, are there product quality data during the production? And they said between brackets after induction. Rui, Julia?
Indeed. I can take that one. So related to the previous question, in terms of protein quality assessment, the experiment or the analytical method that was used, at least the main one, was mass spec, and we have a time-resolved data set for a subset of the fermentations 4, if I'm not mistaken. So indeed, we have several time points of MS data for the same subset of bioprocesses.
Yeah. Someone is asking, has the E. coli strain been engineered by GSK?
Yes, it has been engineered by GSK, but again, the mutations that have been introduced are fairly minor, so they affect mostly secondary metabolism, some regulation, and we are talking about well, less than 20 single point mutations.
Okay. Also, another question, by when would the results have to be shown? How long should the project run roughly? Maybe Julia can make it brief, answer this question briefly.
Yeah, I mean, we briefly answered similar before. We don't have a
Yeah.
specific timeline in mind. Again, we would like to get an update, at least, you know, initial results within a year. But you know, the precise timeline can be negotiated.
Yeah. Also, someone else is asking, which quantities did you measure online? Do we get the preceded data or the raw data plus the calibration information? Rui?
Yeah. So in terms of online measurements, they are the ones that normally come out of well, the equipment. So pH sensors, PO2, these type of well, pressure and of gas. These are normally connected in the control system from the bioreactor, and these can normally be shared with the participants. Other measurements might be a bit more difficult to share, but again, as I mentioned before, in the question regarding the raw data or pre-treated data, regarding which one we would be sharing. Likewise, if there's a strong rationale that is mentioned on the proposal for why we should be sharing one data set rather than the other, we can definitely be a bit flexible on that, provided that the justification is solid.
Yeah. Also, someone else is asking how many batches are profiled for mutation proteins. Rui?
So as I mentioned, the MS dataset was produced for 4 batches, and in terms of mutations, meaning errors that happen in transcription that reflect on the protein sequence, we have transcriptional data for virtually all of the experiments. So for this case, if you are really interested in looking at the mutation rates at the transcription side, we might be able to share with you the FASTQ files. If you are interested in exploring specifically the amino acid misincorporation or the effect of transcriptional errors in translation, then the data set is going to be a bit more limited.
Okay. Also, there is another question. Is the data full and complete, or are there any missing values there? Rui?
Yeah. So it depends on the data set. We carried out a very complex exercise in terms of sampling and pre-treatments of the samples, because depending on the analytical method that we would use, we would need to freeze the cells or to extract directly the metabolites or RNA, and these of course take some time. If we then overlay this with the regular, well, let's say biomass measurements that also take some time for sampling and preparation, well, it's a complex exercise. All of this to say that the data set is, of course, is not aligned in terms of time points. So there's some hours in terms that separate the different samples to allow us to pre-process the samples that we take, taking into account the analytical method that we are going to use to characterize a particular sample. And on top of that, we also have to take into account the working hours of our colleagues that are working in the lab. So there's some length of hours where our colleagues were sleeping. So traditionally at GSK, we don't do shifts at night unless we have a strong reason for that. So there's this gap in terms of characterization of the bioreactor.
Yeah, thank you, Rui. Also, another one, someone is asking about the plasmid's DNA. So was the plasmid's DNA also sequenced to check whether the plasmid was replicated correctly?
Yeah, certainly. So there's so on in terms of sequencing of DNA, I can already give some additional information. So the E. coli strain was resequenced really in the beginning of this project, and we checked, we confirmed that the genetic background of this strain is stable. In terms of plasmid DNA, we didn't characterize the plasmid DNA throughout the bioreactor or in several of the bioreactors. It was a limited number, so 2-3 samples that we use, and that I don't know if we'll be sharing in these data sets. But again, if the proposal really requires it, we can consider it. But answering specifically your question, the sequence of the plasmid was also stable, and the plasmid was present in enough copy numbers. So that's also another thing that we assess for the resistant marker.
Yeah, and just to clarify, the MS data consists only of the target proteins or the full E. coli proteome. Rui?
So I think I mentioned that briefly, but in any case, to clarify, there was a minor protein purification. So the conditions of this purification we can also share with the selected participants. And the reason for it was to be able to eliminate most of the host cell proteins, because for the overall objective of this project, it was not interesting. It was not the scientific question that we were asking. And on top of that, when I mentioned minor purification, the reason for this mild purification was to not eliminate some of the truncated proteins, given that we would like to characterize and to understand what type of errors are going on in E. coli in these processes.
Yeah, thank you, Rui. Thank you for making that clear. There's another question asking, does GSK prefer for academic organizations or for companies to apply? Julia?
We have no preference. Academics, companies, collaborations between the two, all of these are fine. The only restrictions, just for compliance reasons from GSK's side, we cannot accept proposals from healthcare providers that prescribe GSK products, and we cannot accept proposals from government officials that could influence GSK's business. And if you are worried that you might be one of those, go check the terms and conditions. There's a full description there. I guess the other thing might be if you are a direct competitor of GSK, if you work for, Moderna, then we might have to get clear. We might have to clear that through some upper layers of our management to make sure that's okay. But otherwise, we really don't have a preference on what kinds of organizations submit.
Yeah, thanks, Julia. Or someone else is asking, are collaborations between academics and small companies okay? Stephen?
Yeah, just to follow up on what Julia said, we love collaborations between different organizations, particularly where the two organizations are bringing different skills that complement one another, and where 1 + 1 makes more than 2. So we welcome collaborations between academics, between companies and academics, particularly where that adds extra things to the capabilities.
Yeah, thank you, Stephen. And since we're at the end, Julia, one last question. Are there restrictions on how we can use the prize money, like for travel, equipment, team payroll, etc.?
Nope, you can do whatever you want with the money.
Okay, thanks. Thank you, Julia. So we're at the end of the Q&A, but don't worry, if you already sent in your questions, we'll get back to you. So that's no problem. So if we missed any questions that have been asked in the chat box, we will get back to you through that. And thank you for everyone, and thank you for engaging with this Q&A. Very interesting questions. And as I stated previously, any questions not answered here live will be available along with the recording of this webinar, and also for also on the GSK Biomanufacturing Challenge page on 9sites following this webinar. So what you can do today is you visit the challenge page. So you go to 9sigma.com/s/JSK bio dash MANUFACTURINJ so 9sigma JSK Biomanufacturing Challenge. To read the description of the data set, you can find that on 9sigma link. So you go to 9sigma.com/S1A. And if you need any help, you can ask the 9sigma Solution Provider Help Desk at the email phd@9sigma.com. Also, there is a direct phone. You can call us on the number, and you have the extension number 3. You submit your proposal, and the deadline for submissions are Thursday, October 14th, 2021 at 5 p.m. EDT time and 11 p.m. CEST time. Also, that concludes our webinar for today. One final reminder that this webinar was recorded, and it will be made available along with the different questions that have been asked in a transcript. Yeah, and thank you to our speakers today. And thank you for your time and your expertise, and to our audience today for engaging. And we really appreciate your time and look forward to receiving your submission soon. Have a great day, everyone. Thank you.